Hi, and welcome to another episode of STEIN Fireside Chat. We're here today with Dr. Sean Pittock, who is the chair of neurology at Mayo Clinic and a neuroimmunologist, and Dr. Nathan Staff, who is a neuromuscular neurologist and the chair of the Mayo Neurology Research Committee. And so thanks for coming on today. Hope we'll have a nice chat. We'll give you your steins.
Thank you very much.
You can keep that.
Thank you. Appreciate it. Great to be here.
We'd like to talk a little bit just about how you kind of got interested in medicine and then got interested, I guess, in the research aspects.
Well, as you probably know from my accent, I'm from Dublin, Ireland. I've been in Minnesota for 26 years, came over in 1999. I really started off with a passionate interest in psychiatry, actually. I was reading a little bit of Freud when I was in high school, got really into it, and I always decided that I was going to be a psychiatrist. So in Ireland, you go straight from high school straight into medicine, and I started medicine at University College Dublin. Really loved it and loved my psychiatry courses, but when I stepped into the psychiatric hospital to do my rotations, it was at that point that I suddenly realized that psychiatry was not for me, neurology was for me. And so I ended up in neurology, very happy that I continued in that vein. So that's how I ended up in neurology. In Ireland, you go straight from medical school into either medicine or surgery. And your internship, you have to do half your time in surgery and half your time in medicine. So I did 6 months of urology as a surgical intern, admitting 26 patients for urological surgical procedures in one day, and realized very quickly that urology was not for me. So then did internal medicine, went into neurology. Didn't really feel I was getting a good education in Ireland. It's very much you learn from your mistakes on your patients, whereas I really was looking for a didactic kind of course where, a place where I could be taught and learn better. And so interviewed throughout the US and decided that Mayo Clinic was the place that I wanted to do my training, and I started my internship here in 1999, so many years ago, and haven't looked back. Really enjoyed it. Love Mayo.
At what point did you know you wanted to go into MS neuroimmunology?
Yeah, well, that's a story. So I did, when I was doing neurology in Ireland, I decided I was going to do stroke. At that time, TPA was coming out. It was kind of somewhat exciting. And, I also was wanting to get away from the armchair neurologist concept. Didn't like that. I'm much more of a gung-ho guy. And so I thought, yeah, I can do some interventional kind of work here. But when I came here, started to work and learn in the residency program at Mayo, I became interested just by being a resident and watching and learning from the staff that autoimmune neurology or neuroimmunology was of interest. And so I knocked on Vanda Lennon's door in her lab and said, "Hi, I'm a second-year resident. Could I do some projects with you?" And she said, "Yep, come in." And I started off that way. She gave me a small project to do on a rare paraneoplastic disease, which I did, and I did well. And then she gave me another project. And then I happened to be very, very lucky. I was in the lab working with her at the time of the discovery of the water channel antibody, which is the first ever antibody biomarker of an MS-like illness. And I helped out in that project, managed to get an authorship on that discovery paper, and I worked on that disease for 25 years and still working on it.
Got it. Well, Nathan, tell us a little bit about your journey as far as how you got interested in medicine. And
Yeah, I mean, a lot less ocean story than what Sean had to provide. I'm a Midwest person, so I grew up in Wisconsin, went to the University of Minnesota for undergrad. Always liked biology growing up, always digging in the dirt, playing in the fields, playing with bugs. Got to college and decided that I liked neuro things, kind of also reading books about the mind and neurophysiology and things like that.
Where was college?
At University of Minnesota. Wanted to start doing research, interested in trying to decide whether to do a PhD or MD. Ended up in a retinal physiology lab and did work with some of the first confocal microscopes and did some electrophysiology, impaling retinal ganglia cells, which was a lot of fun. And that kind of combined with interest in ultimately having something practical led me to MD PhD program. I did that at Northwestern, really enjoyed that. Spent 4.5 years doing my PhD. I did patch clamp neurophysiology in slices of rat hippocampus. So our kind of claim to fame in that laboratory, which was Nelson Sprucen's laboratory, would be to patch upon the soma of a pyramidal neuron and then also on the long apical dendrite, 300 microns away from the cell body, and look at the conductances and voltage changes that would happen throughout that cell and correlate that with learning and memory sort of phenomenology such as long-term potentiation. And at that point, trying to figure out, I knew I was going to, I finished up medical school, knew I was going to do residency, wasn't sure whether I was going to do neurology, neurosurgery, neuropsychiatry, neuroradiology. Didn't really like my neurology hospital experience. So I was leaning toward neuroradiology, and then I did family medicine, and that got me back to neurology because I really liked the outpatient setting and the relationships with patients, getting to know them. And then residency, it was, we had a small kid at the time, so we're wanted to stay close to family. My wife's from the Twin Cities, so we applied in the Midwest. And Mayo was by and far away the best program at that time, and still. So ended up coming to Mayo for training and ended up getting interested in, thought I was going to go do MS. You may have recalled I was hopping around saying hello to all the MS people. I even did an MS project during residency. But then I did EMG, and it brought me back to physiology and the electrode and the electricity, and I was hooked. So I switched over to neuromuscular and haven't looked back since then.
So both you guys actually measure EMGs. You do it in movement, you do it in neuromuscular disorders. To the neurosurgeon, in my mind, this is like the same thing, but I know that for you guys, it's very different. Could you explain to us sort of how the types of tests that you do with patients are different from one another, and then the kinds of functions that you're both looking at?
So for neuromuscular EMG, and he can talk about this, it's a very detailed and kind of a smaller region that you're looking at a time. For what I do with EMG, it's really either surface EMG or we do some needle EMG polygraphy.
Can you explain what you mean by surface EMG?
So with the surface EMG, you're recording the muscle's electrical activity with stickers. So basically, a surface electrode is kind of like an EKG electrode positioned over the muscle. And then a needle EMG would be you just one of the electrodes at the tip of the needle and you put that into the muscle, and so you get a higher fidelity recording. But either way, what I'm more interested in is the activity in multiple EMG channels at once. So for example, in tremor, you may see a contraction of an agonist and antagonist in a certain pattern together, or in dystonia. Whereas, I'm looking at the overall pattern and the overall flow between the muscles. Whereas in your case, you're looking at a single motor unit.
Yeah, and as you probably remember, we went through clinical neurophysiology at the same time.
We were co-residents and did the EMG
training at the same time.
Yeah.
Diverged a little bit. But yeah, so I mean, there's two main parts of the EMG study. There's the nerve conduction study where we shock nerves and record potentials either over a traveling wave through a sensory nerve or kind of magnified through the voltage changes that happen when a muscle contracts. And we get information about, the strength of the fidelity of the axon. Is it a full axon response, or is it slowed in some way in its conduction, which gives us insights as to damage to the axons or the myelin within the nerve. And then the second part of the test is, as Bryan was mentioning, you stick a needle electrode directly into a muscle. And there we can get down to the resolution of single myofibrils and activity that are happening within that. That can tell us a little more detail about, is there damage to the nerve? Is there damage to the muscle? And as you know, every muscle is innervated by different nerves, so it can kind of tell us where in the nervous system the problem might be. Is it a length-dependent process where things are bad out in the feet and get normal more proximally into the thighs? Or is it something at the root level where, say, for example, the fifth lumbar nerve root innervates a muscle down here, but also a muscle in your hip. And so you can kind of tease apart by innervating and examining all those muscles where the problem is.
I see. So obviously on this podcast, we're fairly interested in prosthetic devices of different kinds, measuring electrophysiology with new technologies. I guess from my perspective, I would think, well, a lot of what you do is you're trying to understand diseases of the brain, and sometimes I guess of the brainstem and spinal cord, but mostly of the brain, and then using signals from the muscles to understand those diseases, and then we have various interventions that we do. Whereas for you, the abnormalities may arise really anywhere from the brain down to the periphery. Is that right?
Yeah, absolutely. So, a lot of what EMG and nerve conduction studies are looking for are problems either at the anterior horn cell or the dorsal root ganglion out into the periphery neuromuscular junction for diseases like myasthenia gravis, or the muscle itself for like muscular dystrophies and myopathies.
And do you foresee a time when we'll have technologies where we're using recordings that we make from the brain at the same time as we're making recordings from nerves and muscle, and basically trying to assist normal biology in the disease state with prosthetic devices that are really distributed throughout the whole neuroaxis, beginning up at the brain, but actually ending at the end effector muscles? Is that the kind of thing that will happen, or do you think, you and I, of course, do some work with neuroprosthetics together, and I don't know that that's the best thing for today's conversation, but in terms of neurotechnologies, is there a role for devices that are hybridized between the brain and the periphery?
Absolutely. I mean, I think it will ultimately depend on where the major part of degeneration is occurring. So if the nerve's gone and the muscle's gone, then you're going to have to, have some sort of prosthetic that's, more of a cyborg or robotics control that may be being controlled central nervous system or at the spinal cord level. If there's issues where the nerves and muscles are still intact, but the signals from the brain to the nerves and muscles aren't working, say with a spinal cord injury, potentially you could utilize the continued living muscle and nerve to be able to have an effector through a brain interface.
Well, Nathan, I was kind of interested to hear how things have changed since we were in EMG lab together, really qualitatively, I guess, assessing, sizes of these units and these patterns, and how, whether it's algorithms or actually new technology, has really changed the field of, I guess, EMG nerve conduction.
Yeah, I mean, as you may know, the actual physiology and basic technology really hasn't changed much since the 50s. It's just we have computers to drive it much more sophisticated fashion than we had previously. I do think that we're right now at kind of a critical juncture where algorithms and the computers are fast enough to actually run the algorithms on the data as it's coming through during a patient study, to where we can actually be able to kind of utilize that technology. And so, we've done a fair amount at Mayo Clinic with this. Some things are very nascent programs. We've taken all of the neurophysiology data and we've used machine learning to look at, particular types of nerve conduction studies and looked at them in ways that we've never been able to look at them before, using, wavelet and Fourier transform and be able to pick out things that were never experienced before and relate that to specific diseases, and to be able to differentiate different diseases from each other. We can differentiate ALS from inclusion body myositis, which is a very similar disease. It's not built into the workflow of the patient care at this point, but I think we're now in a position where we could potentially do that, and we're working very hard to figure out how to get the data up, get it to the clinician to be able to use it in real time at the time that we're doing these studies. That's mostly been done on the nerve conduction study side, which is a fairly easy time series to work with. Moving over to the needle EMG, which is a free-running, noisy signal, is more complicated. But we've got some ideas there as well that we can incorporate into the practice.
Yeah, I remember even something as simple as the duration of the motor unit. And when I was doing it, it was just by ear, and then you could try to hold a unit long enough to kind of measure it. Are there even algorithms that can do that more easily? If
Yeah, so
to take account where you're at in relative distance to the unit so that you can hold it a little easier. And
yeah, so there are programs within the EMG suite that are able to kind of determine whether a spike has a sufficient rise time to be able to be interpreted, and then to be able to measure it actually at the time. Frankly, we still use our ears for most of it.
yeah, that was going to be my next question, is actually this is all exciting, but actually what's happening in there right now. And that's kind of the same in the DBSO, where we have a lot of tools that we're developing, and I think will be sort of the future. But still, if all the screens were down, we'd be pretty good.
Yeah. I think what I'm really excited about is to be able to pull information out of the data streams that we didn't know was there.
Yeah.
And to be able to utilize that, rather than kind of just making it easier for anyone off the street to do an EMG, because that's not as important, I think.
And in your case, can you tell us about your clinical practice? I mean, you have our interactions are always, and in fact, I guess most of our interactions with you are about your role as a leader. But what does your clinical practice look like in your role as a clinician?
Yeah, so my clinical practice really evolved. So when I started at Mayo as a resident and then fellow, there was no field of autoimmune neurology. So there was neuroimmunology, and neuroimmunology was multiple sclerosis. But and a lot of people think when you say inflammatory brain disease, they think multiple sclerosis. Well, you can imagine there's a whole world of diseases that affect both the central nervous system, the brain, the spinal cord, but also the peripheral nervous system, that are immune mediated, that are not multiple sclerosis. And so during my fellowship, we kind of created a fellowship for what we considered a new field of disease, where you actually have immune mediated attack of the nervous system, but different to multiple sclerosis, where it's immune mediated, but we don't know what the target is. This is a field where we study immune-mediated diseases where we actually know what the neural target is. So, for example, in the field of NMO, which is a really severe form of multiple sclerosis, in that condition, in 2004, Van der Linden led the team that discovered the first ever biomarker of a disease, where we actually showed that the disease was caused by an antibody that targets a water channel that sits on an astrocyte. And that was a seminal advance, because that led to the first FDA-approved drug for any inflammatory brain disease other than MS. And this was all done at Mayo Clinic, so this was a seismic jump. So that was happening in the lab, and then the work trying to figure out the mechanisms by which that antibody causes the disease, with the ultimate objective of finding a novel therapy and cure. That was all happening in the research realm. Whereas in the clinic, we developed the first autoimmune neurology clinic in 2007. We developed the first autoimmune neurology fellowship in 2008. And actually, we founded the first the autoimmune neurology section at the American Academy of Neurology in 2017. So the clinic essentially attracted patients that came with inflammatory brain diseases that were not multiple sclerosis. And though each individual disease was rare, when you put them all together, they're actually not so uncommon. And they're complex diseases, because you can imagine that you've got a brain which has, 200 billion neural cells. Most of them are astrocytes. And then you have the immune system, which has an incredible diversity of potential antigenic targets, with B cells with antibodies that target millions of different epitopes, T cells with T cell receptors that target, and so you mix those together, you've got high complexity of disease.
Sure.
And so what we did was, through the Center for MS and Autoimmune Neurology, and through really having, first of all, recruiting and training brilliant people and developing incredible teams, we began to see the patients, but then enroll them in biobanks, biorepositories. And so unusual disease, we would then collect their blood, their spinal fluid. And then, working in conjunction with the neuroimmunology laboratory, we began to search for biomarkers like aquaporin-4 antibodies in NMOSD. And how did we discover that antibody? Well, we took people's blood, we took their serum, we put it on mouse brain tissues, and we looked for immunoglobulins, IgGs, that were binding to the neural targets on the mouse tissues. And when we looked at, for the water channel, for example, we saw a pattern of staining. And then that pattern of staining, we used a variety of different techniques in the laboratory. There was older techniques back in the 90s. Now we have much more technologically advanced techniques. And that allowed us to discover what the target of that antibody was. And at the current time, we're discovering about 2 to 3 novel antibody biomarkers of disease every year based on those patterns of staining. And each of those antibodies has a very, very clear clinical significance. So these are antibodies that they're not just research hypothesis testing antibodies. These are real clinically relevant antibodies. And so the clinic essentially grew with this kind of back and forth from the clinic to the lab, discovering, identifying a target. Once you've figured out the target, then you figure out the disease mechanisms, the immunopathogenic mechanisms. And that then leads to, novel targets. And so then we moved into trials and then have been leading many, many trials looking at novel drugs that target the mechanisms that are involved in those specific, very defined diseases, and ultimately leading to significantly improved outcomes for patients with very, very bizarre inflammatory brain diseases.
So if you have some person come to you with an as yet undescribed but clearly autoimmune fluctuates, there may be triggers that you can identify, disease-causing neurologic disorders. They come in, you may or may not find abnormalities in their MRI that point to there being a problem. Right now, and for individual people, you're trying to discover new therapies?
Yes.
Or is it that you cluster people and then look for a therapy within that?
So you can cluster people in varied ways. So for example, one of the ways we cluster the people is based on the staining pattern. So patient comes in with a phenotype, you find a staining pattern of the immunoglobulin on that mouse tissue. And then a year later, you see another patient with a similar phenotype, and lo and behold, they have the exact same IgG that's binding to the mouse tissue in the same pattern. So you cluster them there. Once you identify more than 5 patients with a similar phenotype, and they all have the same IgG biomarker, the next step then is to figure out, well, what does that IgG bind to? What's its target? Once you've found out the target, then you have a novel biomarker of that specific disease. Then you take that IgG into the lab and you try to figure out how does it cause pathogenesis? How does it injure the cell? Once you figure out the pathways by which it injures the cell, then you've identified potential novel targets.
Sure. And
So
you guys have done this?
Many times, yeah. So for example, in the water channel, what happened was we realized, and this was work even before the discovery of the target, Claudio Lucanetti had shown that in the immunopathological studies of people that had died of this very rare form of MS, in the lesions there was complement deposition. So indicating that this was probably an antibody-mediated disease and complement activating. But once we figured out the antibody that targets the water channel on the astrocyte, we did a variety of different studies led by Shannon Hinson and Van der Lennen that demonstrated that when the antibody binds to the water channel, it activates complement and it kills the cell. So then in 2012, we did a phase 2 trial of a complement inhibitor, and it was dramatic. And then we did a phase 3 trial and showed that we could reduce the likelihood, by blocking complement, we could reduce the likelihood of a clinical relapse of that disease by 96%.
Okay.
So, that's an incredible impact for a disease where these patients become blind or they develop paraplegia because they get attacks of optic neuritis or attacks of inflammation of the spinal cord. To come in and develop a drug based on the immunopathogenic mechanism that came from those discoveries and end up with a drug that reduces the chances of an attack by 96%, essentially nearly moves the patient into near cure. And so then another disease, MOGAD, we've just demonstrated the early results look very positive for an alternative target approach there.
What's MOGAD?
So MOGAD is, so if you can imagine that NMO, this disease with relapsing attacks of optic nerve inflammation and spinal cord inflammation that causes people to go blind and paraplegic, that's caused by an antibody that targets a water channel on an astrocyte. Well, you can just imagine, well, you've got billions of astrocytes with billions of targets, and you've got lots and lots of B cells. So it's not rocket science to imagine that there's another disease out there. And the next disease then in the field of inflammatory central nervous system demyelination other than MS was MOGAD, where the antibody is targeting myelin oligodendrocyte glycoprotein on the oligodendrocyte.
Okay.
And similar disease, patients get optic nerve disease, they get spinal cord disease, they look like MS, but they have antibodies that are targeting the MOG protein on the oligodendrocyte. And in fact, they activate complement. And their mechanism of injury to the cell is very, very similar to water channel antibody positive disease. And so similar drugs are now being tried in that disease as were tried in NMOSD.
Okay. It's kind of interesting. Have you guys ever tried to take these diseases and refashion them for immunotherapies for things like brain tumors? Or, I hear oligo and I'm a simple man. I think oligodendroglioma, take it out, right?
I love the idea. So we have thought about that. So essentially, there's a couple of things to think about. So the first thing is we suspect that people that have tumors that express neural targets that then develop neurological disease because they develop an immune response to the tumor, but the target antigen in the tumor is a neural protein. And now there's a T cell response against the tumor, say in the lung, but now the immune system is now also being directed up to the brain tissue where that neural antigen is also expressed. That's a paraneoplastic neurological disease.
Sure.
And what we noted was that patients with those diseases, they actually generally, we think, do better from their lung cancer perspective because they have a very robust T cell response to their lung cancer. And we also know that in the concept of cancer, if you use a checkpoint inhibitor for the treatment of cancer, you can actually make the brain disease a lot worse. So the idea that you could actually use the response to the neural proteins in a tumor and actually develop potential CARs, then you could actually treat tumors better. So in other words, you're really essentially creating an autoimmune neurological disorder to kind of kill the cancer. And I think that's a very reasonable thing to think about.
Okay.
The other thing is on the other side of that is at the moment we're using CAR T therapies, okay, to take out.
Could you, for our audience, just explain what a CAR T therapy is?
Yeah. So CAR is a chimeric antigen receptor. So essentially it's like having a GPS system in your car. So you're driving along in your car, okay, and you don't know where you're going. So you put in your GPS system, you put in the address, and it takes you right to where you need to go.
Yep.
So this is basically taking a T cell, so from the individual themselves, and putting in that GPS system, the CAR, into their cell. That T cell will then go to wherever that CAR tells it to go. Okay? So, for an example, hematologic malignancies where you have abnormal growth of B cells, so lymphoma, you can now take a T cell from that person, put in a CAR that will target a protein that's on the B cells. And then so you take the T cells out of the patient, you transduce them, viral vectors or whatever, you put the CAR in, grow them, pop them back in the patient. Those CAR T cells now will go and kill every single B cell that expresses the protein that's targeted by that CAR. And that has been revolutionary for people that are essentially have failed all therapies for hematologic malignancy. Suddenly now people are being cured. Well, you can imagine that you could use that in an immune-mediated disease, okay? So there's been work done in people that are dying from lupus, bad lupus. You go in and take out their CD19 positive B cells, these patients are going from being nearly at the point of death to making significant recovery. The same in the field where the immune system is targeting the brain. In our field, we know what the target is. So we have just, Anastasia Zakharadu has just helped in a trial of CD19 CAR T for stiff person syndrome, which is a disorder where you have an immune-mediated target of your inhibitory nervous system and you get very hyperexcitable patients that are in severe spasms and pain, very, very disabled, and it seems to work. But the concept of when you know the target of the immune cell, then you could actually develop a CAR that would just take out the B cell that's expressing the antibody that's targeting that specific target. Or you could potentially develop a CAR that would just take out the cytotoxic T cell based on that T cell receptor phenotype. So now I'm talking about what could happen in 5 to 10 years. So at the moment we have crude CARs that'll just take out a lot of B cells or a lot of T cells. But the future for the field of immunology and CAR T, I think, will allow us to develop much more specific opportunities to just take out the bad ones, the bad immune cells, and that could have significant impact for the field.
Thanks. Something I think that's interesting is when people talk about, I mean, you're one of these rare individuals that have had some scientific finding in the lab and identified patients whom that was relevant for, and then a therapy that saved the same patients that you ultimately came to treat clinically. You know, there's a vision of somebody who happens to notice something and they're basically geeks for the subject, and that's great, right? And that they kind of just follow something doggedly over years and are able to make that whole journey, although I think in practice most people don't have the wide skill set that's needed. Separately, there are also these large teams of individuals where it's kind of like a Manhattan Project for a particular disease model or a particular type of pathology that then results in a therapy. And it seems like you're describing, you've talked about these different individuals that you've worked with, like Dr. Lucianetti, as if they are all sort of different groups that are working together. And so it sounds like the way that you guys have done this is that you have maybe, and correct me if I'm wrong, but some kind of balkanized system where people all interact with one another but have their own specializations, and then you're able to kind of pass tokens back and forth of knowledge that ultimately have led to treating these different types of diseases. Is that right?
Yeah, no, I mean, I just love the way you just phrased that question, because at the end of the day, the first thing that happened was back at the early discovery was what we did at Mayo was we created a Mayo Clinic NMO enterprise, or a group of individuals that all had interests but all came to the disease from a different perspective. So for example, you had Brian Weinschenker and Dean Wingerchuk, who were the diagnostic criteria people. They were the epidemiological people. They studied the epidemiology of the disease. You had Claudia Lucianetti, who studied the immunopathology of the disease. You had Vanda Lennon, who was an immunologist, a basic science immunologist, who discovered the pattern and ultimately the antibody. You had me, who was kind of bringing the lab and the clinic together and then potentially driving forward with trials. You had PhD scientists trying to understand the mechanisms by which the disease ultimately causes injury. And regardless of the fact that all of those individuals were all come together as a team, it still took 25 years to go from the biomarker discovery to an FDA-approved near cure. Getting at your question, what I really like doing as a chair is the opportunity to say, we have the brains at Mayo, but the question is, how do we create teams around problems and do it in less than 25 years?
Yeah.
Okay? So, for example, with respect to the work that you do and Bryan and your teams do, it became very evident to me very, very early on that this was a scenario where we have at Mayo Clinic an incredible group of individuals with the potential to make seismic advances in the field of neuromodulation. But the way that I would see of doing that and accelerating that is really to create an infrastructure within which that can really happen. And I think that that's what we really need to do. What happened in neuroimmunology at Mayo was we had a lot of people working in their labs with their own technicians, with their own fellows, everyone was doing great work. But then what happened was, through the Center for MS Neuroimmunology, we tried to bring everybody into kind of the same fold. So for example, on a Wednesday, every Wednesday, I meet with every research fellow in neuroimmunology, even if they're not my research fellow.
Still do.
Yeah, still do. Just to know what they're doing. And really to try to break down the walls of labs so that actually people are now sharing lab space. there's a free flow of fellows, residents, PhD students kind of moving throughout the whole program, as well as data scientists who are coming in. And rather than a data scientist working in one person's lab and then another data scientist working in another person's lab, to try and create a core of data scientists that are all working together and that can kind of move fluidly throughout. I think that will allow dramatic acceleration in the program that has been developed at Mayo. As you know, Dr. Geller raised the day when she came as neurosurgical chair, and
my boss,
your boss, and when we met the first time, there was a very clear realization that in this field of neuromodulation, we have an opportunity to not necessarily make anybody specifically better, but potentially create the opportunity for rapid acceleration.
Yeah,
that's what's really exciting, because it's all well and good to say we brought something from a discovery to a very good treatment, but it's still 25 years of your life.
Yeah.
I mean, we should be really moving quicker. And the other thing that's very evident, I think, is the incredible enthusiasm and the desire to move things rapidly.
Sure.
But in order to move things rapidly, you need to have team science, infrastructure, and funding.
Yeah. Well, so coming to the subject of funding and its role in research, obviously there's some challenges with the NIH right now. Hopefully by the time this airs, those will be resolved, but they may or may not be. But the example that you gave where you're talking about neuromyelitis optica, this really doesn't affect a lot of people as a percentage of the general population. And yet you had a team of people where they were all working on it, and they had some kind of support within the same institution to study and treat, ultimately to look epidemiologically at a relatively narrow indication. And because that was understood very well, it sounds like there are a lot of general principles that have allowed for a lot of other therapies. And something that I wonder about, and something that seems to be a challenge, is something I would hope we could do at Mayo, is to identify a few great model situations. ALS is another one. Even though they affect people relatively rarely as a percentage of the general population, there are certain aspects of them that may allow us to have very general therapies. But it's hard to make those arguments when you're going to funding agencies where they say, well, the impact of that compared with, say, epilepsy affects much larger percentage of the population. Now, the problem there, of course, is that epilepsy is a remarkably heterogeneous disease, right? There are many, many, many different types of things that cause epilepsy. And when you're talking to funding agencies and you're talking to leadership and you're talking to potential donors, you run into this problem of, when you're trying to describe the impact, it's always comparing it against what are perceived as very large diseases, even though they may have relatively very varied and very heterogeneous underlying mechanisms. It's just hard to make that case for the well-controlled, because a well-controlled clinical milieu to have scientific discoveries in actually is, in many ways, a better situation as a clinician scientist to make an impact, because you can constrain the problem to a set of essential scientific questions you can address, a set of strategies that are going to be fairly similar across that population of people. And it's something I think we certainly hope to leverage at Mayo, is the fact that people will come here from a lot of different places because we concentrate expertise in things that are relatively focused.
Yeah, I suppose what I would say to that would be, first of all, I think what Mayo does great is we see the rare, we know the rare,
yeah,
and we're very good at helping the rare.
Yeah.
But I would say that what we really are talking about here is the power of rare. And that's why the very reason why you need to have an infrastructure built around the program. Mm-hmm. Because within the program, sometimes all you're doing is rare disease. But when you put all the rare diseases together, they're not so rare. Mm-hmm. In fact, if you look at the diseases that people are coming to Mayo Clinic with, I guarantee you, if you add up all the rares, they will make up a significant proportion of the patients. Now, to give you an example, NMOSD is rare, but it was thought to be very, very rare. And then when we figured out and discovered the biomarker, we could do biomarker-based, population-based studies. And what did we find? It wasn't so rare. So what we did was we took every patient in Olmsted County that had any form of inflammatory brain disease, and we tested them for the water channel antibody, which happens to be 100% sensitive and 100% specific. Then we went to the French West Indies, where a neurologist happens to just travel around seeing his patients and collecting bloods on every single patient he sees, because he was a forward-thinking guy, just on his own. And he had collected bloods on every patient with an inflammatory brain disease on the island of Martinique. And these are Afro-Caribbeans. And we tested them all for water channel antibody. And we found that in Afro-Caribbeans, 10 patients in every 100,000 had the disease. Okay? So you then start to study the disease in Africans, and you find out that actually it's about 4 times more common in Africans than in whites. And globally, there's probably about 500,000 people with the disease. Then you use the infrastructure and the know-how that you learned from that disease to get to a treatment, and you apply it to what? Well, to MOGAD. MOGAD is a newer disease, but we have just completed a phase 3 trial that potentially we hope is going to show positive results. And so now we're beginning to apply the know-how of one rare disease to another rare disease, and what you're going to start seeing is that the rate or the time that it takes to go from the discovery to the cure or a treatment is going to start dropping and dropping and dropping. The same with these very unusual pediatric genetic diseases. I mean, what we need is we need an infrastructure around very rare genetic diseases.
Sure.
So that we can have a pipeline of patients that are coming in with rare disease X, pipeline of N of 1 gene therapeutics, and as you build that infrastructure, you'll be able to do that. And also the concept of repurposing. I mean, you're putting an electrode into a patient that has epilepsy. You're assisting with the electrode implantation for movement disorders, Parkinson's disease, whatever. But you know that once you've got that smoothly running, and it's just now we just do it on everybody that has that disease, then what you're going to do is you're going to start saying, well, what happens if I put that into a patient that has insomnia?
Sure.
What happens if I put that into a patient that has a neurodegenerative disease? What happens if I put that patient into a patient with a dystonia? Suddenly now you're into the rare, but you're using a fix that you're very familiar with, and then you start to rapidly and accelerate, I think, treatment options for those types of patients.
And and don't let me ask all the questions. I'm sorry, but as the leaders of neurology and research within neurology and innovation in sort of clinical neurological sciences here, to what degree do you feel that things should be top-down, meaning you identify priorities that you feel that the institution should have within your sphere of influence, versus waiting for interesting opportunities to come bottom up from the scientists and clinicians here at Mayo? What does that balance look like for you guys as a team?
Yeah, I mean, I think that is always kind of a tension in leadership, because I think many of the most brilliant ideas and brilliant innovations are coming from the bottom up. People who have deep content expertise and have been studying it for years and can make the breakthroughs and bring them through. I think one thing that Mayo does very well is that because there's kind of the ecosystem of basic scientists, translational scientists, clinicians, clinical trialists, that I think we're, as it was shown with NMO, there's a way to very quickly bring things to the clinic once the discoveries are made. But that still is a bottom-up approach. Mm-hmm. I mean, I do think that when there are scenarios where we clearly have strengths that need to be amplified, that need to be accelerated, that's where I think the top-down approach comes better, where, for example, with neuromodulation and the Bionic Initiative, we have a key number of people and groups who've been working on their own, kind of hustling and doing great work on their own, and leadership saw that would go much faster if we were able to put the infrastructure around it and give it the funding and get it moving all together and working together for a singular purpose. And maybe spend a little less time hustling on your own and maybe focusing the spear a little bit more directly toward the future. But I think you can only do that if you have the people there to do it already. So, obviously with neuromodulation, it's been decades of hiring the right people, training the right people, building the teams to where it's now ready to go full tilt. You can't just kind of come in and say, all right, here, we need to become the world's expert in X, Y, or Z, and we don't have anybody to do it. It takes a while to move that forward.
I will say, getting Keith Kaufman, who went from having some pediatric dystonia patients to having many, and being able to leverage expertise by sort of poaching that one key individual. And
No, I really agree with what Nathan said there. I think it becomes very evident when you're buried in a program of research, the pieces of the puzzle that are missing. At the end of the day, you have to build your program around the people that you have, because it's the people that do all the work. But you can be in a scenario where you are missing a piece. I'll give you an example. In the world that we were working in neuroimmunology, it was evident that we were missing a B-cell expert. And so what we did was we went out and we recruited Suresh Irani from Oxford, who is the head of neuroimmunology at Oxford. And we recruited him, but he wasn't willing to come to Minnesota because he wanted the warmth. And so we recruited him to Jacksonville, Florida, where he's doing great. And we interact with him on a monthly basis. We have blood samples and samples going up and down, and combined papers and research papers and collaboration. And so I think, that's where leadership can, I think, help, because there's great stuff going on, but it can become very evident that if we had that cog in the watch, then the watch would work perfectly and everybody would benefit. One of the things that I really dislike about the potential of leadership is the tendency sometimes for leaders to decide on what they think is best for people and then try to force a circle into a square.
Sure.
That's highly problematic. And so I think that, and I think you'd agree with me, that the job of research leadership is to first of all identify the strengths of the individuals in the department, to have a broad vision for where we want to go in the future, and figure out who in the department is aligned with that vision, and then try to figure out what is it that they need to do what they need to do quicker. Mm-hmm. Because for me, we have a lot of really, really brilliant people, but we need to accelerate. And it's why do we want to accelerate? Because I'm sick of sitting in the room with patients who have bad disease and not being able to provide them with what I would want if I was sitting there. And so there is an anxiety and a kind of a need to move quicker. That's what I think leadership has to do. We have to make sure that the individuals that are working in the department are working and aligned with the vision. And the vision for our department in neurology is very simple. It's connect to cure. Anything that moves a patient towards a better treatment outcome is priority. And actually, then if you go through each individual in the department, you'll find that everybody is connected to cure in some way. And the question is just, how do you augment that? And there are many ways of doing that, but it's a tough job. Would you agree?
It has its challenges. We have a lot of good people. I think the other part is trying to foster junior faculty and provide the protection to have the brilliant thoughts in the development of the program, whether that's time protection, whether that's financial protection, and be fairly equal initially, and then kind of as people start to kind of show how they're going to be successful, start to kind of move the chips around and try to accelerate the people who are showing that they kind of have the fire in the belly to move things forward quicker.
How about And I kind of, in my own way, think of myself like this, and certain other people that I may know as folks who are driven by problems that they come into in the course of their clinical practice, and they're at home developing some solution. Like they might develop a new software program for the EMG lab and movement, like Bryan has. But at the end of the day, they don't want to lead a large multidisciplinary initiative and have people below them. What they want to do is they're driven for their passion for the patient, and they want to be the person who identifies some particular problem that they have, and then target that one thing. But the problem that you run into is that you want to empower those people to, and I feel like I'm using catchword after catchword, right? But you want to empower those people to leverage this moment of insight that they may have on an intermittent basis because they're passionate about the substance of the clinical questions that they have, and applying the their unique tools, right? So the way that they think about operating ham radio may help them come up with a better way to program a DBS device. But how do you have a system that works reliably as a department, where you have people that are consistently delivering clinical care, but then in an intermittent fashion they may need more time to do that key thing, and they're not trying to do maybe something that's much larger and much different than they otherwise did. I'm very much grateful and in awe of people that are in leadership like you guys are, but I wouldn't want that myself, because it's just not what motivates me. But if you had some situation where you're supposed to be pushed into that in order to go to that next step, because that's the standard mold that in many institutions leaders see and funding agencies support. But if you look at the history of breakthroughs in science and medicine, they are often made by the people who, their goal isn't to make breakthroughs in science and medicine. Their goal is to take care of patients, and they happen to come across something that's particularly interesting. And then they need the time and the resources to focus on that. How do you guys make that happen, right? How do you empower the unpredictable?
I think that's a really good question. I don't think there's a perfect answer, because there are many examples of like Nobel laureates who just were surprised by their success, and it's very difficult because we're in a situation where we are kind of wanting the researcher to kind of know what the future holds, which is very difficult to do. So how do you manage that? You've got this great idea, you want time to pursue it, but it is possible that that great idea potentially may not be as good an idea as you think it might be, or it might be the next Nobel Prize winner. You don't know. That's why I think the benefit of having a structured, interactive program is really helpful. Because it's very helpful to have somebody go, I think that's a really bad idea.
Yeah.
Or, holy cow, I've never thought about that. That's brilliant.
Yeah.
Depending on the response that you get from others, that sometimes can help things move along. It's very hard to know which horse to back. There are many people, I've certainly come across many people in my career who thought they were really onto a big thing, and they weren't. And they spent a lot of their careers following something that didn't ultimately pan out. And then you meet people who have, I think, discovered things or developed novel technologies that now we're actually using in our clinical practice. And generally, I would say those individuals are people that are very interactive, very collaborative, and have a lot of pots on the boil and don't have all their eggs in one basket.
Sure.
So I don't know. I think that's a difficult situation. I do think that there are many brilliant people, I think, who can become siloed and kind of attached to their own ideas. And that's why for those types of individuals, I think they probably benefit very well from getting input from others that are honest to try and help them move forward. But, I'm sure that, it's highly possible that, the people that discovered DNA might have gone into their buddy next door and said, what do you think of this? They went, oh, I wouldn't spend time on that. Yeah, I think it's hard to find that balance. I don't think there's a good answer to the question, but I do think if you step back and say generally, in terms of things that ultimately become successful, I think it's generally done in a multidisciplinary, collaborative environment where you have expertise in many, many different areas, all kind of coming together around a problem. I don't know. What do you think?
No, I think that's absolutely critical. But the other thing I think is that there needs to be kind of a system of communication that comes up to leadership. So, you're talking about, which horse to bet on.
Yeah.
If you don't know what the horses are, you don't know how to bet. And so, having, maybe, what we have in the department is we have some very low-level seed funding that is very easy to get. You don't have to write an R01 or much of anything to get resources to help with statistics or coordinator time or even a little bit of your own time. And then, then there's a little higher level where you have to maybe write a page or two. And then it keeps going up as you get kind of more invested in it. And each of those levels, that has to come up through leadership for us to ultimately say yes. So at least we know kind of what is going on in the department. And other ways of communication are through the divisions and through to the chairs that way. And then I think the other part is to have an institutional infrastructure in place to be able to capitalize on ideas quickly. So, having some sort of, intellectual property, Mayo Clinic ventures, things that are hopefully nimble and able to, respond to your ideas and be able to either resource them or at least, kind of give you the path forward that you may not be interested in yourself.
How about, and this is maybe the tougher question, how about the opposite? How about people that you have come in who market themselves from the start as, let's say, clinician scientists, and at the end of the day, after 5 years, you find out that what they're great is they're great at coming in and marketing themselves as clinician scientists. They can tell you a great personal story, and every time it's a personal story that's inspirational, but at the end of the day, they're maybe taking care of a lot fewer patients, and they really aren't into the substance of the thing that the science that's related to the clinical care they deliver is about. What they're into is their own story, but they built themselves up this way, and that's their identity within the department. But they can still be very good doctors, but that's the narrative that's associated with them. The right term shouldn't be like a graceful declination of the degree to which they fit that label. But is there a way to make it so that, in particular, I think institutions may identify folks who come up through residency, and out of residency, they're supposed to be the next big thing. But what they were good at is a sales pitch. But at the end of the day, they're not staying up late at night thinking about the oddity in that patient they saw that day, and how that may be related to, like you said, to some other disease that had some similarities, and maybe a similar approach could be taken. What they're doing is they're invested in something that has nothing to do with their practice. So they built themselves up with a great personal story, and I think that there's a lot of that. And I think as clinician scientists, this is always something that they essentially train people to do now. But there should also be room, while you want to empower some people that aren't doing as much, are there mechanisms to sort of bring things down for the folks that are commanding maybe a lot of resources, but not necessarily having that commitment to the dual role that was sort of the plan when they started?
Well, I would have to say, just to cut in here, that fortunately we don't have that problem in neurology. I don't know what Kai's talking about. He must have.
I don't have any particular people in mind.
He must have something that he knows about.
I'm not.
I totally agree with you.
Nothing subtle here. Nothing subtle here.
We'll cut that part.
We don't have to cut that part. I think.
Let's talk about another department.
Yes.
But I'd be happy to weigh in on that one. What I will say to that is I think that everybody is different. And everybody brings something to the table. I think individuals who, speak to speak, if you want to go that way, at the end of the day, it becomes clear at the end of the day what's happening. And so whether that individual goes back to seeing patients or whatever, generally, you'll find that people over the years is ultimately figure out where they best lie, where they're best fit. There are a lot of people that started off thinking I'm going to be a clinician scientist, and now they're running trials, and they love running trials, and they're very happy they don't have to worry about the stress of getting an NIH grant. There are people that I would consider really, really brilliant scientists who cannot get NIH funding. Why? Because they're out of the box thinkers. They want to go and do it and accelerate things, and let's just go do this. They're probably right, but National Institute of Health is not interested in that. I think it can be difficult, and I think one of the jobs, I think, of leadership is to try and help people figure out where they best lie, where are their talents. I mean, if they're employed at Mayo, they're good. I mean, they're going to be excellent.
Well, I think that. I mean,
so. Yeah. I think we try to do that. From a research perspective, I mean, Nathan spends a lot of his time helping people navigate research at the institutional level, at the national level, from a space perspective, from an advice perspective. You try to help people with respect to that. Also helping and developing a structured mentorship program, which we've done. So that the younger investigators who kind of can get lost. I mean, it's all well and good. You finish your residency and you think, we know this. You interview people for residency, and they're talking, when I'm going to work 80-20. I'm going to get 2 grants. I'm going to get a K. I'm going to, there is a sudden reality of it's actually very hard to get an NIH grant. But at the end of the day, some people then realize that actually, I'm happy. I'm not going to spend 6 months of my life not seeing my family writing a grant where I suddenly get a 18%, which is better than 82% of the other people, but I'm not going to get funded. I mean, that can be hard.
Yeah.
I just think that if an individual is in that scenario, hopefully they need to get some good advice to try and help them find the best place for them for their careers.
Yeah. So I was at a meeting and ran into somebody, let's say very high, I don't want to say who they are, very high up in the NIH that I've known for many years, who basically told me in this environment, with things as they are, you have a 5% or less chance of getting a grant funded. Maybe you should wait a little while before you kind of re-enter the grant game. And that's the person who their job has been to, and previously they were always telling me, oh yeah, you should submit your next thing, you should submit your next thing. And now they're saying, well, I don't know how these things are going to recover or if they're going to recover. I'm not necessarily asking for an opinion about that, but for you guys, what's the plan if this model that's existed in the post-World War II era of government funding through meritorious, and you could argue what the merit is, but if this environment doesn't exist anymore, how are institutions that do biomedical research, how are their scientists going to be able to keep functioning? How is innovation going to keep happening? Is it going to have to all move over to the industry side? And if so, should medical institutions partner with industry? I mean, usually for very good ethical reasons, there's been a line there. What happens?
Yeah, I mean, for me, a couple of things. I think, first of all, if people are doing good work, then they should be supported. And don't underestimate the quality of the work that can come from low-level funding. Yeah. In fact, I would say that a lot of the best discoveries and work has come at Mayo Clinic has come from not hugely funded entities. Mm-hmm. Sometimes the funding comes after the major discovery is made. Mm-hmm. But I do think that, and I think Mayo has done a good job of this, is diversification. Greg Gores, the last chair of head of research at Mayo, was very good and had foresight and diversified the portfolio. But yes, I think, at the current time, it would appear that NIH funding is going to drop. We don't know by how much. But I still think that good research will continue to exist, and the people that are doing it will continue to get support. But I agree, it will be through potentially different mechanisms. And those mechanisms will be, people that really believe in the disease and want to accelerate it and are willing to provide, benefactor funding through development, etc. Closer interaction with industry partners, and visionary startups and companies that see the vision. So, for example, the work that you're doing, I don't think it takes a rocket scientist to know that having artificial intelligence assess and develop algorithmic approaches to predicting what electrical data coming out from the brain is going to have an impact. I mean, we know that this is going to be big. And so maybe the way to do this is different. It's to say to a company that, there is a huge future in this. And maybe you need to decide to put some of the dollars that you have into working with Mayo or working with other institutions to accelerate the science, so that at the end of the day, because at the end of the day, the drugs or the devices that are going to impact patients are not going to come from you guys and the Mayo Clinic on its own. Sure. It has to have that commercial relationship. And I think that the way we do science is and the way we fund science is changing. Mm-hmm. Yeah, I don't know, what do you think?
Yeah, I think diversification is obviously the key. I mean, things are just very uncertain right now. I think, fortunately, it looks like, in 2026, the funding for NIH is going to be stable, maybe even a little bit improved, which is obviously a much different scenario than we were talking about a year ago. But I think it's really been a wake-up call for institutions on how to manage potentially a drastically cut NIH system. And it's not just the dollars that actually do the research. From the institutional standpoint, it actually is more frightening the indirect support of the F&A fees that come with that, with the NIH dollars that basically fund most of the infrastructure that we have to do the science here. We need to have regulatory oversight of what's done both at the patient level, the clinical level, device level, giving us infrastructure to be able to move things through the FDA correctly. I mean, 40% of the indirect dollars that come into Mayo Clinic actually end up paying to keep the lights on, literally, the buildings and the infrastructure there. So obviously those dollars have to come from somewhere to keep the lights on. I think Mayo's fortunately been in a very good position as being a very strong clinical practice to be able to fund other aspects of our mission. I think other institutions are going to be in a much more difficult place because they're much more reliant on those dollars.
Something I'd really like to ask you guys about is you both wear, at least 3 hats. So professionally, and then of course you have your outside lives as well. But you're functioning as clinicians, functioning as scientists, and also functioning as leaders. How do you take the things that made you good as scientists, so your own initiative? I think for all of us, we are at least in part fueled by our own egos. I certainly am. I think that's normal. How do you put some of that to the side in order to function as a leader and try to promote beyond yourselves? And do you find that difficult? I mean, maybe some people are natural at that, but are you guys natural at that? Or what's the thought process behind sort of the things that have always been your sphere of personal development and the intellectual contributions that you make as an individual? Or has it transitioned, or did you just add something different on as you now are trying to really fuel a whole set of individuals and enable them to sort of pursue the things that they feel sort of intellectually and personally are important for them to do, and much of your role as leaders must be to help make that happen. So where does that trade-off lie? I mean, for most people, they can't turn the scientist button off in their head. You come home, and for Dora and I, this is like a conversation. Well, I just thought of this idea. It just doesn't go off. But for you guys, I mean, you really now have to adopt this role where you are sort of fueling the curiosity of others. How does that work?
I suppose for me, the leadership roles that I've really had before taking on the chair role were really more laboratory based. They were more around science. And I don't know if I would necessarily say leadership. I mean, we were a team of people who were passionate around science and around moving a kind of a new field forward. And I really enjoyed that. My favorite part of being a fellow and a young consultant was the opportunity to get into the weeds of research and really kind of, take a concept and move it forward. I think eventually you get to a point where you've basically, I think, mentored people that essentially are perfectly capable of doing what you had done and probably are doing it better. I think novel technologies come in and then allow quite a dramatic acceleration in the opportunities to discover. So for example, some of the things we were talking about earlier, we now have opportunities to use phage display where you can essentially screen a serum sample very, very quickly for the ability for that antibody to target any protein in the entire human proteome using millions of phages that have little pieces of human DNA in them that are expressing the little pieces of the entire human proteome. And we've used that to discover an antibody biomarker in collaboration with UCSF. But that was actually led by Div Dubé, who then took that technology from UCSF and the Zuckerberg Biohub and brought it to Mayo. Now you're discovering using a mechanism, a methodology that's been brought in by somebody who was a fellow and is now running that program themselves. And the same happens to a lot of other technologies that come into the lab. So at some point you kind of sit back and say, all of this discovery is now being done by other people. The program is vibrant. And yes, at one point there was Vanda and myself, and now there's Vanda, myself, and loads of other people. And so the program is robust, and that's what I love. I'm very happy to step back and just see the vibrant productivity of individuals who at one point were fellows. They're doing great. So then the question is, how do you live with the fact that it's not your baby? You're not, it's not your thing anymore. I don't have a problem with that. I still like to talk science. I like the idea of having ideas, but I have no problem with having the idea, sharing it with somebody and letting them run with it. Now, in terms of, switching from the science to the, to leadership, I don't know. I tend to lead by just, group effort, team effort. And I think, there's just really, in our neurology department, we've got really, really brilliant people. And I don't know if there's a lot of leadership. I mean, I think that it's hard to lead really brilliant people. They don't really need a lot of leadership. What I prefer to do, though, is to think about ways in which we can really develop novel programs around problems and identify, again, like we were saying earlier, the pieces of the clogs in that watch that are required to make that happen and move it forward and accelerate it. And I take a lot of pleasure in that. It does require stepping back and letting essentially other people do it. But that's fine. I mean, at some point, you do evolve. But it doesn't mean that I'm not passionate about science. So maybe, I'm not the site PI that's enrolling the patient. Maybe I'm sitting on an advisory committee for a drug company and saying, maybe that's the wrong design for the trial. It's just a different scene, but I still enjoy it. I don't know. What about you?
Yeah, I mean, I think for me, the things, I still have kind of like the science brain that I need to it's the itch that always needs scratching. But I do that kind of compartmentalized in some ways from a lot of the other stuff that I do. I mean, it's compartmentalized many times during the day, so it keeps coming back in. But I think really what I have always enjoyed, and I think what led me into leadership, is finding synergies and win-wins. I've always been a team player. I love team sports. I love kind of the idea of that, groups are better than the sum of their individuals. And so, I think as I've been in this role and I've been at this institution long enough, and I know enough things that are going on, that I feel like I just want to be able to share that to accelerate other people's work. Like what what office is here, what person is there, what lab is there, and try to bring people to when they have questions and they feel like they're in a roadblock, to get them moving quicker and not feel like they're kind of lost in a sea of bureaucracy. Because everything's at Mayo, you just need to know how to get to it. And so a lot of what I do is to try to do that. But I also love the idea, I think, as you said, is, coming up with the idea and the direction, say, okay, we need to go in this direction from a research standpoint. I may not know all of the methodologies and all of the rigorous science that needs to get us there, but I know that you do, and I know that you do, and aren't we all excited about this specific disease and to be able to come up, say, with treatments for ALS, which is a horrible disease and we have no great treatments for, and get people excited about trying to break through and come up with new therapies for that, and push them all in the right direction and let them flourish in the ideas that they have, but always kind of keep bringing in the context and also, what's going on in the field and being able to kind of provide more of a guidance and steering of the ship rather than, the details on the ground. I have things where I have control of all the details. It's the little science brain in me, but I also recognize that there's some huge unmet needs that I have skill set to be able to try to move the field forward, even if I'm not the one doing the basic science myself.
And then you also are a small business owner, right? Can you tell us a little bit about that?
Yeah, it's small business owner is probably a stretch for me specifically, but my wife is deeply involved in the coffee shops that we have in town called Cafe STEAM. There's three owner groups.
Picture over here.
Yeah, absolutely. We started that 11 years ago. Put a lot of elbow grease in at the beginning of it. It was successful because we have, again, great people and allowed them to kind of have their vision flourish. And I mostly just sat back and drank coffee and commented on things. And now we're opening our fourth one up this spring.
Yeah, I'm excited about this. Right across from the hospital.
Yeah, absolutely.
But I think in the intellectual world, coffee shops hold a special place. The intellectual breakthroughs of the late Enlightenment period came from, the idea of salons and all this. This was coffee houses.
Yeah,
it wasn't bars, right? It was,
yeah.
And certainly for me, all of my graduate work was in Seattle. Working in coffee shops was where I did my best math.
We didn't feel like we had that in Rochester when we moved here. So this was the drive because just like you, graduate school, medical school, undergrad, I spent hours and hours and hours in coffee shops, either, reading by myself or arguing with people about ideas. I mean, it's a place for that. And there really wasn't that. And I think that we do serve that function, particularly in the original coffee house.
I spend a lot of time arguing with people in your coffee
shop.
Yeah,
thank you. I appreciate that.
Yell at my children a little bit, whatever. We really want to see the field of neuromodulation grow and flourish. And obviously we're interviewing you guys here from Rochester, but it's sort of part of a larger initiative.
This is going to be a
You know what I would love?
This is going to be a movie. This is going to be a 1.5 hour movie.
It's going to take 30 seconds.
You need to cut this down.
30 seconds of my speaking.
This is movie part 1.
Yeah.
We're coming back for movie part 2. All right. I have to run, man.
Thank you for coming.